Hey everybody. So it's about time we did an update video on the Rangpur Lime and finger lime chimera, or at least what we strongly suspect to be a chimera, a periclinal chimera, where one of the layers is potentially Rangpur lime and the other layers are finger lime. There's a lot of history behind this, a lot of data we've been able to collect so far, and as of now we officially do have the, let's call it the first round of results of laboratory testing that we're having done on these plants, specifically the suspected chimera and the controls being the actual giant finger lime that was the original Siam variety, and the Rangpur lime, which was the intended rootstock. So we recently had these tested with flow cytometry to essentially get some gauge of their genome size to see if there's any difference in that, or pretty much any other differences, different ploidy, a different number of chromosomes, or if there's anything else we'd be able to tell visually from the data that was being collected. So I actually have a lot to share with you guys. I'm going to try and consolidate it in this video and not go on too much about stuff that we've already talked about with these, but you could probably tell, just right in front of us here, we have three different plants here. We're going to focus on these first two primarily, and you could tell while these are both phenotypically finger limes, one of them is clearly different from the other. And can you spot which one that is? That's the one on the left here. This one on the left is our suspected chimera because it has behaved differently from all the other finger lime grafts that we've done since day one. In fact, even before day one, because this began growing before any of the other grafts did, which is a point that we're going to get to. Well, heck, let's even start with it, because I want to go over all the supporting facts that we have for this potentially being a chimera, and of course going into the actual lab results and everything, and just kind of building a case for you guys so you can see my understanding and my thought process behind this and where it's taking us, because it's really interesting and I'm learning a lot, and I think a lot of this information can be used for the synthesizing new chimeras, and ideally combinations that would be preferable for cold hardiness and edibility and things of that nature. I want to use it as some, not necessarily as a path for breeding, but I want to essentially get a lot of high quality citrus varieties and improve their hardiness to a degree where we can grow them in areas where our selection's a lot more limited. So yuzu is one of my primary focuses that I'm hoping I can use the information I'm collecting here to create some sort of yuzu chimera, because it's already very hardy, but if we can get one even hardier, that would be a great start. And then from there, it's limitless, the amount of combinations. It's truly up to your imagination to some degree, but we'll go over what makes some of these combinations more ideal as well, because there are some factors to consider with, again, the origin of this. So let's get into that. When I grafted the finger limes onto the Rangpur lime rootstocks here, it was done in the winter time, which for me was very convenient because we didn't have a lot of other nursery work going on. But in the same sense, it was not ideal because the grafts would heal slowly and they would begin to grow even slower. So one of the issues we saw was that finger lime has a higher heat requirement to start growing than the Rangpur lime have. So what we had was a lot of suckering from the Rangpur lime and no growth from the finger lime. This resulted in a lot of upkeep and a lot of maintenance to get these plants eventually to grow the finger lime portion that we were desiring from those grafts. I had a lot of extra budwood from our giant finger lime, which I had top worked with blood lime, which is a hybrid, funny enough, between Rangpur lime and finger lime. So I had all this extra budwood and I had all these extra Rangpur lime seedlings, and it seemed like a pretty compatible combination to me. I couldn't find a lot in the literature about it, but I do know, the same species, basically, Citrus limonea, lemons. So the Volkamer lemon is a rootstock that's used for finger limes that I believe behaves somewhat similarly to Rangpur lime, though it might be a bit more vigorous. Finger limes actually do great on Volkamer lemon, so I figured on Rangpur lime they ought to do pretty well too. And I actually have some more thoughts relating to that and how the Chimera, this combination, could be a really, really good one. But again, the origin of this, so we had all this suckering occurring, the grafts weren't growing, and as we were getting closer and closer to eventually reaching those ideal temperatures, I did notice, and I've done some close-ups here before, I did notice, and you can kind of still see it there, a shoot coming from the graft union of one of these plants, and it was growing with the suckers when none of the other finger lime shoots were growing. Now this was suspicious to me because, eventually one shoot has to be the first to start growing, but why was this one the first to start growing? The one that was in the middle of the graft union. And on top of that, I've taken photos of this too, but it even had a distorted look to it. When it began growing, it was very unusual and twisted. The leaves looked a bit different, and the new growth didn't look quite like the finger lime growth that I was used to on the giant finger lime. It's normally very densely clustered and dark red. It has a lot of those anthocyanins, and this one simply did not have it. And that's a trait we'll get into that I've kind of noticed as it's continued growing along. But we had this very suspicious shoot that started growing when the Rangpur Lime shoots were growing. Now this was also weird because we had sliced into one of the buds on that particular scion when doing our side veneer graft, and that healed with the callus tissue, and from that bud is where this shoot came. So this bud may have, and I do indeed think this happened, I think it intermingled with the callus tissue of the Rangpur Lime, which then suckered and almost had like some sort of exterior shell of the other layers being the finger lime, which is a really interesting combination and just a crazy phenomenon to even think about. But the fact that this happens because of the different heat requirements, I mentioned this may be a factor to consider when thinking about other graft chimera situations. I think it's possible no matter the time of year. However, under these specific circumstances, I think I actually created very ideal conditions. I grafted over 100 plants, so the odds were in my favor, but the actual number that I sliced, particularly like this, must have been a dozen or less. So to have like 1 in a dozen in terms of my success rate, that's really good. And I think the circumstances, it was definitely partly because of that. So it might be ideal to have a rootstock actually that has lower heat requirements to start growing than the scion variety, or just one that's more prone to suckering, whereas the scion variety might be a little slower. So I do know sometimes, not always, this time of year yuzu grafts actually grow great, but when it is cooler, yuzu might be more hesitant to grow, whereas a Citramello, Citrange rootstock, even a Citrandrin like X639, those might be more inclined to sprout out at that time of year. So a combination like that might be ideal if you could start getting that suckering happening before the scion is actually growing. And of course, trimming back any scion growth that isn't at that bud you sliced, that would be ideal for kind of controlling it and really trying to focus that energy where you want it. So I am replicating this accident currently, although temperatures are different. I am doing the same exact thing, and I'm just cutting the buds off the scion, cutting any suckers off of the rootstock unless they are coming from that graft union, and more particularly from that bud that's being sliced, because that's how it happened the first time, so I'm going to stick to the method that worked. But now let's go into plant phenotype, how these plants appear, what are the overall differences. So before we get into the lab results, I want to point out the obvious in what I've noticed. So as you can see here, the plant on the left, these plants were originally in the greenhouse. I moved them to our patio actually, so I could do some closer observations and some microscope stuff with them, which I also saw some very interesting things I'll put up in a minute. But as you can see, under those different light conditions, the growth on this regular finger lime was relatively consistent. You know, the leaves maybe changed a little bit towards the top, got a bit more ovoid, a bit more serrated, but nothing too crazy. Very typical finger lime habit there. Fairly consistent. But under this change of conditions, our suspected chimera here, just look at that up close. Look at those long internodes. And that's not the only thing. The leaves are much, much bigger, and the veins are more profound. So you could probably see that up close. You see how noticeable those veins are? I mean, and when I've compared these plants up close, it's a noticeable difference. But it takes a little bit of time to manifest, and it's not always consistent. It depends on light conditions and nutrient and all sorts of things. So I do believe at some point, though, we will see a more stable character with this. We'll just call it a chimera. I jokingly called it a Lim-era because it's a Rangpur Lime and a finger lime chimera, but we'll just call it that for the time being. But you could just see this chimera is really distinguishing itself. And we're going to get to why I believe these specific differences are important, because it may further evidence which of the layers actually got swapped with the Rangpur Lime. So just taking it all into account, it's generally more vigorous, it seems. It has longer internodes. It has larger leaves that are more serrated, and have more profound veining. And something I noticed too, which you can kind of see there, is it has petioles, and much larger petioles on some of these leaves, whereas the actual finger lime tends not to have any at all. And this is one of those moments where I'm going to point at the screen here. I'll pull up a close-up from the microscope of those leaves compared side by side, and you can see structurally they're very different. Towards the base of the leaves, the chimera really kind of points in, and it has that additional wider petiole where it's much more rounded off with hardly any petiole with the actual finger lime. So that's a substantial difference. And another thing I've noticed, depending on the growth stage, is reduced anthocyanins in the new growth. It's a little harder to see at the top here on these long shoots that we have, but generally speaking, the chimera has had less of a reddish tint in that new growth. And we can actually see here it's been putting out a new shoot here where you could see that as well. It's not super reddish, not what you'd expect from a finger lime. Just look at those leaves too. Those are some unusual shaped leaves and very long internodes for a new shoot on a finger lime. Because I do have a control here too. Look, that's one I found. That's a new shoot on a finger lime. That's typical. That's what we'd expect, that dark red, that really clustered look. And that is just so different from what we're seeing here. So again, it's not incredibly obvious when you just look at these, but when you really break down the details, there's some substantial differences between these two. It's just not immediately obvious. Now, we'll get into it, but I do suspect that L3, so there's 3 layers we're essentially looking at here, L1, L2, and L3, and they do have, the plant's broken up into different components, so the vascular system is primarily L3, and, in terms of fruit quality, what we're kind of more interested in, L1 and L2 dictate that more so in terms of the internal fruit, which I believe is L1, and L2 is probably more like the pith and the peel and things like that. So I'm not entirely sure about the influence on the fruit. At most, we might just have larger fruit on this, and that's it. Because if the actual L2 doesn't have any Rangpur Lime influence, certainly I don't think L1 is Rangpur Lime. So if we're narrowing it down, I'm not expecting much, if any, difference in fruit, but more so in terms of growth qualities and ease of propagation and things like that. And we'll talk about that a little more, but I want to get into the lab results. So the flow cytometry that we had done, I'm going to put up here the graphs comparing these. You're going to see graphs of the Lim-era, the giant finger lime, the Rangpur Lime, and you'll see these side by side, and you'll notice they actually are different. So in terms of genome size, the genome between Lim-era and the giant finger lime were about the same. In fact, the Lim-era came in slightly lower, but it wasn't significant. So statistically speaking, they were probably about the same. And the Rangpur Lime was, whereas these were like a 0.79, 0.8, the Rangpur Lime genome size came in at like a 0.84. So it did seem potentially significantly bigger. So there was a difference in that, but more importantly too, and I'll put up here too, I had the scatter plots and all the data overlaid between the Rangpur Lime and the giant finger lime and compared it to the Lim-era. And although the genome size indicated, well, the Lim-era and the giant finger lime were about the same, that's not what the raw data and the histograms and the scatter plots, that's not what any of that said. When I overlaid it, it looked so similar between the two. It was crazy. It opened my eyes up completely, because this was something I wasn't able to see just from the actual result of the test. But when we're looking at the data, you could see, again, very, very similar in terms of like the number of events or the number of cells being counted and where they're clustered together on those scatter plots and on that plot of the raw data. There's even a point where, I don't know if I could even circle it, there's like a little cluster that was shared between the Rangpur lime and the Lim-era that was not present in the giant finger lime, at least not to any significant degree. So I noticed similarities shared between the Lim-era and the Rangpur lime that weren't present with the giant finger lime, despite you would expect it to look, well, just about exactly the same, or at least very, very close. You know, we'll account for some error between tests, but I mean, the fact that I saw a drastic difference that was corrected by overlaying the graphs of the results really said something. And it, again, gave us a strong indication that perhaps we do have a chimera on our hands. Now, I was kind of tinkering around with the idea, and I even used AI to help me out a little bit. Why would the genome size be the same or potentially slightly smaller than the Rangpur lime if it has that additional Rangpur lime DNA going on? You know, I'm no geneticist. I don't know all about this stuff. But it seems to me, because I did leaf samples, and you can see actually there is a leaf missing here, and I took one from there and all that. The leaf samples, based on the contribution of the different layers, so L1, L2, and L3, I believe L3 is underrepresented in the leaves. The vascular system is not a strong majority there. It's kind of outnumbered, basically. So it doesn't have as much of a presence as you would otherwise expect. So I guess that's potentially somewhat flawed in how I went about this. I could have tested stem and leaf tissue, but I opted just for the leaves instead. So still, though, we noticed that difference, and that, I think, was very meaningful with that. But something I want to talk about and think about when it comes to this L3 influence here, or just the fact that if this is a chimera with the Rangpur lime and L3 is the Rangpur lime, well, what does that mean for this plant? What are the differences actually going to be? And well, so far we're noticing part of it already. It's a probably more vigorous, resilient plant that's going to produce more food for itself. It's going to grow faster and potentially yield better. Not to mention, I thought of this too, if this is a chimera with the rootstock, and well, the portion that got swapped with the rootstock is the vascular system, and that does become the root system then, well, this, when propagated by cuttings clonally, which is normally hard for finger limes and they don't grow as well on their own roots, well, it'll have Rangpur lime roots. It's almost like it's self-grafted, like it's built into itself. It has a built-in rootstock, so it should be easier to propagate and have a more resilient root system. And again, if it performs similarly to the Volcom or lemon, that's actually a really, really good combination for this, because that means it has a good, high-quality root system built into a plant that is otherwise normally grafted. But if you think about the economical implications of being able to propagate these readily from cuttings and have them grow vigorously on their own roots, you could probably propagate these a lot quicker and at a lot larger scale with less overall labor than it takes to graft plants. It's a lot easier to grow cuttings than it is to graft plants. So I thought that was actually a substantial advantage. Not to mention, now let's think about the Prague chimera, which is also believed to have the L3 of a trifoliate orange and the other layers of a Satsuma mandarin. It is a hardier plant than the Satsuma mandarin. It does have some other flaws, but it resists the cold better because of that L3, and there's various reasons. There could be some antifreeze proteins and different signaling and all this stuff. It gets complicated. We don't know the exact reason, but we know that when the vascular system is swapped with a hardier plant, the plant as a whole gains cold hardiness. And while the Rangpur Lime theoretically is hardier, and I do believe it is hardier than the finger lime, which is more cold sensitive. And as indicated by growing at cooler temperatures, well, now you have a plant that will grow not only more vigorously, but start growing sooner in the season and also resist the cold better. Now, again, we don't know in terms of dormancy if that could become an issue, but essentially this should be able to push it into a slightly cooler zone. And I don't know the full degree of that. We're so early on, there's still so much to learn about this plant, but we can speculate. We can speculate based on what we know so far, and I think these are very meaningful differences that, while the fruit might not be different on this plant, the growth habit, and again, that propagation, all that stuff, if all of that is improved, this is essentially just an improved version of the giant finger lime, which already is some sort of bud spore or hybrid. I think a lot of people lean towards being a hybrid with a lime. So this could be some super complex thing where it's a hybrid between a finger lime and, say, a key lime or something, and then it's a chimera with a Rangpur lime. So we just got all sorts of limey stuff going on here, which is really crazy to think about. But all in all, I think this could be a very great improvement that happens, again, completely by accident. I mean, this is kind of like our miracle plant. And despite being so into the hardy citrus, I gotta say this has become one of my favorite plants just because of my special attachment to this now. The fact that this, miracle child happened here completely by accident, and now it's potentially giving us ideas and laying the foundation for creating future chimeras, I think is just absolutely valuable. It's really, really cool because while I do think there's a lot of hope in terms of breeding new hardy citrus and just breeding new citrus in general, these chimeras I think hold a lot of potential. And there's pretty limited research. I try and include some on our website in our posts about the chimeras. But there's some pretty cool cases of chimeras between oranges and mandarins and sour oranges and things like that. And it's really cool to see when those fruit characteristics get blended too, which again could happen. There's no guarantee every time I do this it's going to be the same layer distribution, but there's a good chance if it's repeated exactly the same that the L3 at least could be part of that original plant, that rootstock plant. Now, what are the other layers going to be? Well, there's a good chance one of those could also end up being the rootstock, and you could have an inferior fruit. However, you could also argue, just think about it, for example, if we grafted like a 10-degree tangerine onto a swingle, and we got a chimera between the two, and what would it be? L1 would be 10 degree, and L2 is the swingle. You might have a more bitter rinds and pith and things like that, but then have a sweet, delicious inside, which is still an improvement over standard swingle. Or vice versa, maybe it has the swingle interior, but now it has an improved and more usable peel. I mean, there's subtle differences like that where it doesn't have to change it completely, but one component can really make a difference. Because some of these hybrids are just hard to approach for a lot of people. No matter how much we might like it, to actually get these to commercial level quality is pretty difficult. But little changes like that in terms of the sugar content and the aromas and making them a little bit more normal per se, and more expected, more like what people would expect from these citrus, I think that would really help. But not to go off on too much of a tangent here, I just mostly wanted to, again, build a case for you guys and let you in on some of my thinking behind this plant and the direction of it. I'm getting to the point where I'm almost ready to start propagating it. I just want to wait for it to harden off a little bit more and stop growing, because it hasn't stopped growing and it's shooting out more. It's, again, very vigorous compared to its counterpart, which I've had this control. It started sprouting not long after this one, and it's outpacing it by some margin. And again, you can see that difference in leaf shape and size and everything. It's just doing really, really well, but I only have one of these. So it's quite precious, and I do want to propagate it just to have more backups and to really be able to study this more. And I want to see what those fruit end up looking like, what those flowers end up looking like. Because I suppose there's still a chance L2 or one of those could also be Rangpur Lime, but because it is phenotypically a finger lime for the most part. It looks very, very similar. I don't know the odds of any of those other layers being Rangpur Lime. It seems too strongly influenced by the finger lime, but you never know. But I don't have a fully grown Rangpur Lime plant to compare at this size here, but you'd be able to see it much more represents the finger lime here. But just crazy to see all those differences. I can't believe, especially lately, how these leaves have distinguished themselves. And something I didn't point out too, if I didn't show it already, the buds on this plant too, I even noticed the difference in the bud shape. The buds were larger and a little differently sized, just more prominent, more noticeable on the Rangpur Lime Chimera than on the finger lime. The finger lime buds are just very hard to see in general, and it's definitely quite tricky to graft these, as well, as you could tell just from all the thorns and everything. So it's going to be a fun time propagating this one. But anyways, guys, let me know what you're thinking about all this. It's a lot of information all at once. I just got to drop that on you guys. Take a moment to let it soak in, think about it. But I'm excited to hear that some of you are also trying to do this. And ever since I posted about this on the Tropical Fruit Forum, about recreating the Prague Chimera, there's been a lot of people trying. And that trying is what's going to get us closer. A lot of experiments, some might work out great, some might work out bad. It might just not work out at all. But this is how we're going to learn how to do this. And, throughout this time of me trying, and at some points I wasn't able to put as much effort towards this as I wanted to, my first potential success here happens by accident. I wasn't even trying to make it. But I think this is one of those happy little accidents where we're going to learn a lot from this, and that'll be very telling as for where to go from here. How do we make more of these? So, yeah, let me know your thoughts in the comments. Make sure you like and subscribe if you're into this nerdy citrus type of content here. But naturally, I'll keep you guys updated with the progress of this one. I might not update much further until we get some SSR analysis done, because I believe that's a more clear-cut way to tell similar genes and stuff between these. I think it's a lot easier using that method as opposed to the one I started off with. So I'm going to try and get some more definitive testing done on these plants to really be able to distinguish them and say, yes, I'm 98% certain this is a chimera. But, until then, it's just kind of letting this grow, trying to propagate it maybe, and we'll kind of go from there. There's not going to be a whole lot of really meaningful updates until we get those secondary lab tests back, and until we start seeing this thing flower and fruit, and maybe see if we notice any more differences as it continues to grow larger. But that's it for me, guys. I hope you guys enjoy the rest of your week. Again, let me know your thoughts, and I'll see you in the next one. Bye-bye.